Genetic mapping is a technique used to determine the relative positions of genes on a chromosome. It's a fundamental tool in genetics, providing a roadmap for understanding the organization and function of genomes. How does genetic mapping work? The principle behind genetic mapping is based on genetic linkage. Genes that are physically close together on a chromosome are more likely to be inherited together. This is because they are less likely to be separated during the process of recombination, which occurs during meiosis. Crossbreeding researchers crossbreed organisms with different traits. Offspring analysis. The offspring are analyzed to determine which traits are inherited together. Map construction. The frequency of co-inheritance is used to estimate the distance between the genes. Types of genetic maps. Linkage maps. These maps are based on the frequency of recombination between genes. Physical maps. These maps are based on the physical distance between genes measured in base pairs. Applications of genetic mapping. Identifying disease genes. Genetic mapping has been used to identify genes responsible for various genetic disorders. Improving crop yields. By mapping genes associated with traits like disease resistance or higher yield, breeders can develop better crops. Understanding evolutionary relationships. Genetic maps can help trace the evolutionary history of species. Challenges in genetic mapping. Complexity of genomes. Large and complex genomes can make mapping difficult. Recombination rates. Variations in recombination rates can affect mapping accuracy. Genetic heterogeneity. Some traits may be influenced by multiple genes, making mapping more challenging. In essence, genetic mapping provides a valuable tool for understanding the genetic basis of traits and diseases. By mapping genes, scientists can gain insights into their functions and develop strategies for improving human health and agricultural productivity.